Welcome everybody. So today we're going to have two conferences. Pierre Savatier is going to be talking for the first 45 minutes. He's a world specialist in understanding the fundamental biology of stem cells, cells that can be engineered to give cells of interest, which of course are being used in regenerative medicine. And at SPRI, the arrival of Pierre drove us to develop the MPTP model, which is being furthered here by Tremblay and others. And today Pierre is going to be talking about aspects of this kind of approach in terms of producing animals of interest for models of diseases. Thank you.
Thank you. So I think my talk is going to be a bit different from at least some of the other talks, in that I'm not going to speak at length of what I've been doing before, but rather trying to show you which technologies are now available to make transgenic animals and how these technologies could be applied in primates and to generate transgenic monkeys. And this is the title of my talk. Okay. So I want to give you some background and some definitions before starting the actual talk. So what we call genetic modification is also called genetic engineering, and this is direct manipulation of an organism genome using biotechnology. And new DNA can be inserted into the DNA of the host organism, and this is usually called gene addition, and this is one of the most simple technology. But you can also remove a gene, and this is called a gene knockout or gene ablation. And there are more sophisticated technologies which allow genes to be modified to alter their function, and this is usually referred to as gene alteration. Genome modification is achieved by manipulating the genome of the early pre-implantation embryo, first prior to the formation of the germ cells, and therefore any modification that you introduce into the embryo will be transmitted to the offspring. Genome modification has been developed over the past 30-40 years in rodents, and there are recent attempts to transfer these technologies to livestock and more recently to primates, and this is what I'm going to discuss today. And I just want to draw your attention to the point that genome modification is strictly prohibited in human, although the technologies which I'm going to describe could also be applied to human. So why create genome modification in non-human primates? And the main reason is to study gene function, pretty much for the same reason as this has been done in the mouse in rodents over the past 30 years. And in primate, this is interesting to more specifically study the function of genes involved in brain development and brain function. But it also has it has also another major interest, and this is to create monkey models of human genetic diseases, and this to study disease etiology using technologies that cannot be applied to humans, and also to assess the safety and the efficacy of gene and cell therapies, and this is something which is very important at the present time in the context of the so-called regenerative medicine. So the first technology I'm going to describe is actually something that we've done. We have worked on a few years ago in collaboration with the Institute of Zoology in Kunming in the south of China, and this technology is based on the infection of the fertilized eggs of the rhesus macaque with a virus vector carrying foreign DNA. So we used lentiviral vectors. There are different sorts of lentiviral vectors, either vectors derived from the human immunodeficiency virus, or, and this is the case here, we used vectors derived from the monkey, the simian immunodeficiency virus. And these lentiviral vectors have the capacity to integrate any kind of foreign DNA sequence into the genome of the host cells. And we have engineered here a very simple virus carrying the gene here, EGFP, which you probably know, and which expresses the so-called green fluorescent protein. And with this virus, we infected rhesus embryos at different developmental stages, and then further cultured these embryos till the morula or blastocyst stage. I will come back to this developmental stage later on, and then transfer these embryos into surrogate mothers. So this is an example of some pictures of the experiment. So the virus is injected under the zona pellucida here. In this particular example here, the virus has been injected into an 8-cell stage embryo. This is the expression of the green fluorescent protein in a morula stage embryo. As you can see here, not all cells are positive for GFP, which means that the resulting embryo is chimeric for GFP expression. And here, this is a picture of a rare event in rhesus macaque, is the ultrasound imaging of a triple pregnancy, because we used to reimplant three embryos into each surrogate mother. And the results of this experiment is shown on the next 2 slides. So this is some pictures of the newborns we obtained. So this is the 2 newborns here on the bright light, and here under the ultraviolet light, to observe the GFP expression. And as you can see here, there is a transgenic newborn here, and the other one is a negative control. And this one is another transgenic newborn. So we got 2. And this here I showed you some figures which, first of all, the important question is, does the infection with these embryos with the vector interfere with normal development? And the answer is clearly no. In the control experiment, we had 60% of developmental rate of development of 60% and 50% with the embryos after injection with the antiviral vector. So there is no interference with development. And so the second question is, what is the rate of transgenic newborn production? We started the experiments with approximately 200 fertilized oocytes, which we got 30 blastocysts which expressed the green fluorescent proteins. We got 4 newborns, and of these 4 newborns, we got two transgenic animals. So the efficiency is around 1%, which is very low. But the loss in efficiency does not result from the infection. The rate of infection is very high. The loss of efficiency results from the poor developmental capability of the fertilized oocytes in primates. Most of the embryos, actually, most of the fertilized eggs do not develop to the blastocyst stage. The developmental rate is around 15%. Okay, so now I want to show you an example of, I think time is running fast. So this is an example of a work that's been published by the group of Anthony Chan in Atlanta a few years ago, in which they tried to produce a transgenic model of Huntington disease in non-human primate. So just to remind you that the Huntington disease results from the mutation of a specific mutation in the Huntington gene. The Huntington gene is a very large gene, and the mutation is localized in the first exon of the gene. And in the first exon of the gene, there is a stretch of triplets, CAG, which are repeated less than 26 times in the normal gene, in the normal allele, right? And when the number of repeats increases between 27 and 35, this results in a so-called mutable normal allele, which means that there is no disease associated with this gene, but the gene has the capacity to further mutate and increase the size of the stretch of CAG. And this is what happens here in the Huntington gene. So when the number of repeat reaches the threshold of 40, this results in the full disease. And when the number of repeats is between 36 and 39, there is the disease develops, but with an incomplete penetrance, which means that not all individuals develop the disease. So they try to recapitulate the human disease in the monkey, and they injected, using the same strategy that I developed, I explained before. They have injected an antiviral vector expressing this mutant huntingtin carrying from 27 to 88 repeats of the CAG triplet, together with GFP in order to follow the fate of the embryos. And they have been able to produce a number of transgenic animals. These transgenic animals all exhibit aggregates of huntingtin in the cortex and in the striatum, which is the first anatomical evidence of the disease. And here is a table that summarizes the symptoms of the disease in the animal. And so with in one transgenic animal carrying 29 repeats, which therefore is a normal huntingtin gene, there is no movement dysfunction. And in the transgenic animal that carry 83, 84, or 88 repeats, CAG repeats, there are movement dysfunction observed in these monkeys. So I'm not going into the detail of the experiments, just to show you that it is possible to indeed recapitulate some aspects of the human disease in the monkey using a very simple technology that is infecting the embryo with lentiviral vectors expressing the mutant genes. Unfortunately, this is a very rare example of what can be achieved in the field of monkey models, because in the case of the huntingtin gene, the overexpression of the mutant gene is sufficient to induce the disease. But in many other diseases, you have to remove the endogenous genes in order for the disease to develop. And so this is a brief table which summarizes what I just said, that the strategy using the infection with lentiviral vectors is suitable for gene addition, but is not suitable for gene modification or gene ablation. So you have to have other technologies to introduce very subtle mutations into the genome of the monkey. So I'm going to describe another technique which has been developed in rodents and more recently in livestock. And this technology is based on the so-called zinc finger nuclease, which are injected into fertilized eggs and pre-implantation embryos. So what this zinc finger nuclease strategy is, so this is based on an enzyme extracted and purified from a bacteria, and this is called the FOC nuclease. And this is a nuclease which has the capacity to cut the DNA into small pieces. But unfortunately, this FOC nuclease has no specificity, which means that if you introduce the FOC nuclease into the cell, an animal cell, it's going to cut the DNA into small pieces. And so what you need is a strategy to target the nuclease activity towards a single gene in the genome, the gene that you want to mutate. And the strategy that has been developed is described on this slide. The general principle of the strategy is the combination of so-called zinc finger modules. These zinc finger modules are small proteins which have the capacity to bind DNA with sequence specificity. So you have, as it's shown on this slide here, you have 4 different modules, one which can bind specifically to the GAC triplet, another one GTC, another one AGT, another one GC. And by combining these 4 zinc finger modules to the half of the FOC nuclease here, you generate a recombinant protein which has the capacity to recognize the GAC, GTC, AGT, GTA sequence in the genome. Unfortunately, this sequence is not unique in the genome. Statistically speaking, if you look at the sequence of a primate, for instance, you will find that there are many GAC, GTC, AGT, GTA sequences. Therefore, you have to have more specificity, and the extra specificity is provided by the second zinc finger modules with 4 other proteins recognizing 4 other triplets, TAT, GAC, AGT, CGA in this particular example. and fused to the other half of the FOC nucleases. And the result of this is shown here. Now, by using 8 independent zinc figure modules recognizing a sequence of 24 nucleotides, then you achieve the specificity that you want. This zinc figure protein is capable of recognizing one single sequence within the genome. And if you choose these sequencers appropriately, you can recognize and target with very high specificity the gene that you want to mutate in the cell. And then this is how it works. You have the zinc figure module, the left one here, the right one here, and the two parts of the nuclease cut the DNA. And the resulting, so this is shown here. So the zinc figure nuclease is injected into the nucleus of the cell or of the embryo. It binds and cuts the gene that you are targeting. This induces the so-called DSB, which is double strand break. The two strands of the DNA is broken, and then takes place a process called non-homologous end joining. So the two pieces of DNA sticks together, and then this creates a small deletion or small mutation in the genome. So this has been used and developed in the rat, in mouse, in zebrafish, now in rabbits, and in theory, this strategy could be developed in primates. There is no reason why this strategy couldn't be used to target any gene in the genome in the monkey genome. However, this technology is suitable for gene ablation. You can introduce a small mutation into the gene and therefore disrupt the gene function. But there is, it is far more difficult to use this technology for gene addition or gene modification. And I'm not going to describe this in detail, but this is something which is still under development. So now I'm going to talk about pluripotent stem cells, because as I showed you on the previous, on this slide here, so if you want to disrupt gene, we have the technology in hands. The big question now is how we can introduce more subtle mutations into the animal genome. And this, in principle, could be achieved using pluripotent stem cells. So I'm going to give you a brief introduction about the pluripotent stem cells and see how we could use these stem cells to create very sophisticated monkey models of human diseases. So embryonic stem cells, so there are two types of pluripotent stem cells, the so-called embryonic stem cells and the induced pluripotent stem cells. Shinya Yamanaka, who is the scientist who first developed these induced pluripotent stem cells, got the Nobel Prize this year, and therefore this is a very trendy and hot topic. And so to start with the embryonic stem cell, these embryonic stem cells are produced from the early pre-implantation embryo at the so-called blastocyst stage. So there are two parts in the embryo. There is this clump of cell here called the inner cell mass, and this is where the stem cells of the embryo are. So the whole organism derives from this clump of stem cells, and the other cells here form the so-called trophoblast, and this layer of cells will later give rise to the placenta. So the embryonic stem cells can be generated from these blastocysts, and they can be propagated with no limit in culture. So they proliferate. They can proliferate very actively, and these cells can be genetically modified very easily. There are another Nobel Prize actually has been awarded for the development of different technologies for the genetic manipulation of these embryonic stem cells. And these embryonic stem cells are pluripotent, which means that they can differentiate into all the cells that form the body, including neurons. I think it's too much for the computer to have all the things running together. So you have here neuroprecursors and neurons. I'm trying to activate. Okay, anyway. Yeah, you have your liver cells, and here you have cardiac cells. This is the most fascinating, but surely it doesn't. Okay, anyway. So yeah, right. The cells are, so you can have beating cells in the culture dish when the embryonic stem cells are treated with appropriate growth factors and culture conditions. And they can also differentiate into oocytes and sperm, so therefore they have the capacity to make all the gametes and contribute to the germline. The same holds true for induced pluripotent stem cells. So you probably all know that pluripotent stem cells are produced by reprogramming of somatic cells in culture with the so-called Yamanaka's factor, after the name of the scientists who first described this technology. And this, starting with fibroblasts, these pluripotent stem cells arises in the culture, and they self-renew and form bigger and bigger colony, and they also have the capacity to differentiate into all sorts of tissue types, both somatic and germ cells. Okay, so now the question is how to make, a picture is missing here, I don't know why, but this is just a picture of pluripotent stem cells, so it's not so important. So the question now is how to make a mouse from genetically modified pluripotent stem cells. So I'm going to describe very briefly 3 technologies which have been described in mice to create transgenic mice from pluripotent stem cells. The first one, which is of course the most well-known, is the microinjection of ES cells or iPS cells into the mouse blastocyst, and this has been first described in the mid-80s. And the general principle of the experiment is to genetically engineer the ES cells or iPS cells in culture, and then inject the cells back into the blastocyst. And this film shows you how this is performed. Is very quick. So you have the holding pipette, the injection pipette with the cells, and the cells are injected inside the blastocyst. And these cells which have been injected inside the blastocyst colonize the clump of stem cells of the host stem cells, and the ES cells, the injected cells here, contribute to the development of the blastocyst after reimplantation into surrogate mice. So this is an example of, a typical example of what can be achieved with this technology. These are ES cells which have been engineered to express the green fluorescent protein. They are injected into blastocyst. As you can see, they colonize the blastocyst, and then you can obtain a transgenic offspring with a very high efficiency. The second technique is a little bit more difficult, and this is this technology is based on the nuclear transfer of ES or iPS cells into enucleated mouse oocytes. So the first attempt to perform this experiment dates back to the early to 2002, and in this technology, the first step is the removal of the genetic material from the oocyte, and the replacement of this of the oocyte nucleus by the nucleus of a genetically engineered ES cells. So this is shown on Well, anyway, so and the result of this experiment is the production of, again, so if you transfer the nucleus from ES cells expressing the green fluorescent protein, you get embryos which express the green fluorescent protein in all cells of the embryo, and they can be transferred to surrogate mothers. So this technology has been developed in mice, in livestock. The efficiency is very low, much lower than the technology using the injection of ES cells into the blastocysts. But the difference between these two technologies is that here you get a transgenic animal in the first generation. All cells which derived from all cells of the body of the adult organism which derived from this embryo express the transgene or express or harbor the mutation that you have introduced into the cells, as opposed to the previous technique which I described here, in which not all cells in the newborn express or carry the mutation because they mixed with the host cells in the embryo, and therefore you get a chimera in the first generation. So you have to breed the animals and wait until the second generation to get a real transgenic animals. And the third technology that I want to mention is something which has been published very recently, actually 1 month ago, and this is the in vitro differentiation of ES and iPS cells into mature oocytes. So here the general principle of this technique is to create oocytes in vitro from the genetically engineered ES or iPS cells, and then fertilize these in vitro made oocytes to produce eggs and embryos. So this is what has been achieved. This comes from a very nice paper in Science published last month. So the general principle of this technology is the in vitro induction of embryonic stem cells to differentiate into so-called primordial germ cells, like cells. And these primordial germ cells are the precursors of the real germ cells, oocytes or sperm. And in order to allow these primordial germ cells to further differentiate and form real oocytes, they need to form cell contacts with other cell types found in the gonads of the mouse fetuses. And therefore, they dissected the gonads, female gonads from E13 mouse fetuses, removed the endogenous primordial germ cells, and formed aggregates with the primordial germ cells derived from the ES cells. And they were able to form what they called reconstituted ovaries. And in these ovaries, there are follicles, growing follicles, from which oocytes, mature oocytes, can be removed. They can be fertilized with sperm to form eggs, and they can be developed into embryos and then into newborns. Therefore, it is possible to introduce a genetic mutation into the embryonic stem cells here and obtain transgenic newborns after the formation of oocytes in vitro. So these are the three technologies which can be used to generate transgenic animals with pluripotent stem cells. The beauty of these technologies is that they can be used for gene addition, gene modification, gene ablation. Every type of mutation you can think of can be applied with these technologies. There's no limitation in the application of these technologies. The question now is, does it work in non-human primates? And can we make a macaque monkey from pluripotent stem cells? And this is an issue that I've been recently very much interested in, and that we are going to develop in the lab in the future. So the three technologies which I just described, starting with pluripotent stem cells, so namely blastocyst colonization with stem cells, nuclear transfer into enucleated oocytes, and in vitro generation of mature oocytes, None of the three technologies are already available in non-human primates. We have performed some preliminary experiments regarding blastocyst colonization with proton stem cells in the context of our collaboration with China some years ago. These preliminary experiments were unsuccessful. And we also tried to perform some, nuclear transfer into a nucleotide oocyte in the rhesus monkey, but again, these experiments were not successful. This technology, as I said, is very recent, and nobody has yet tried to apply this technology to primates. So the technology that we are going to work on in the lab within the next few years is this technology, the blastocyst colonization with pluripotent stem cells. We don't have the expertise to develop the two other technologies, so we will focus on this strategy, which for a number of reasons. One is because we have strong expertise in the manipulation of pluripotent stem cells, and this technology has proved very efficient in rodents, and therefore we can rely on the past expertise in rodents to develop this technology in non-human primates. Yet, this is not an easy task. This is not a trivial matter. And I'm going to take just the last 2 or 3 minutes of my talk to explain why this is not a trivial matter. In rodents, this is a time scale of the early development in rodents with the fertilization, the activation of the embryonic genome. This is the pre-implantation phase of development, and then implantation takes place right after the formation of the blastocyst, right? In primates, the whole process is delayed. The activation of the embryonic genome is delayed. The formation of the blastocyst is delayed. It takes place at around the 6th day of gestation. And therefore, there is a difference in the development in the two species. The ES cells which are produced from the blastocyst, the mouse blastocyst, are different from the ES cells which can be produced from the primate blastocyst, human blastocyst, or from the rhesus monkey blastocyst. These two cell types have profound biological differences. And for this reason, the monkey ES cells and the human ES cells which are produced from these embryos are not able to contribute to embryonic development as the rodent ES cells do. We do not understand exactly why it is like this, but the explanation probably stems from the difference in the timing of embryo development, and there are also some important differences in the molecular pathways involved in the early steps of development between the two species. So when I was preparing this talk, I tried to think of, because I'm sure you're not familiar with this, neither with embryo development nor with pluripotent stem cells. So I tried to think of to which other biological system we could compare these embryo and these cells, and I thought of the brain development, the brain anatomy in the primate brain structure, and I would like to compare the rodent, the ES cells in rodents, to the cortical precursors in the ventricular zone, and the ES cells of primate origin to cortical precursors in the subventricular zones. They are different. Although these cells share the capacity to differentiate into a large variety of cell types, they are biologically different. And if you inject neural precursors of the subventricular zone into the ventricular zone, I'm not sure that these the cells coming from the subventricular zone will be able to survive and integrate and colonize the ventricular zone because they have different properties. We are exactly in the same situation here. The ES cells from from primates are not capable of colonizing the embryo, and therefore what we have to do, and this will be our work in the next few years, is to create, engineer embryonic stem cells in primates which are equal to the embryonic stem cells of rodents. And therefore we think that when this step has been achieved, then we will be able to create monkey chimeras harboring, carrying mutations. And my last slide is a picture of my team, and in red here the name of the people who will be involved to some extent in the project in the context of the CORTEX consortium. Thank you.
Paper's open to questions. Pierre, from what you were saying, it would seem that perhaps the nuclear transfer into the oocyte would be as difficult in primates, non-human primates and humans, as it is in rodents. How much more difficult is that technology? Because the route you're taking requires mastering a lot of complicated biology before you're in the same position with primates that you are in with rodents. If you were to do the nuclear transfer, would it be possibly more difficult to do that in primates than it is in rodents?
There is no reason to think it's going to be more difficult. It's only a matter of number of animals involved and the number of experiments to perform. The efficiency of this is reproductive cloning, and the efficiency of reproductive cloning is very low. The efficiency varies from one species to the other. It is relatively low in rodents. It is a bit higher in bovine. So far nobody has ever succeeded in creating cloned animals in primates, in monkeys. But the number of animals that have been, well, number of experiments performed is relatively low. So there is no reason why it wouldn't work, but it would cost a fortune to perform these sort of experiments. That's the main reason why I think this is not something which is which is feasible in the context of.
One of you people out there?
I was wondering whether what type of species you're going to use in primates, and if there are some species that are closer to the rodent type of development, or if this is an issue or not.
No, we had this discussion in the lab, and so there are two opinions. One would be to work with lower primates, and therefore we would be able to have large amount of embryos. The limiting factor in these experiments is the amount of embryos available, and therefore, so this is why these technologies have been developed in rodents. We are doing some, as you know, we are doing some experiments in rabbits, and this is fairly easy because these are species which you can get 20, 30 embryos from a single female. This is obviously not the case in primates. So therefore, with lower primates, we might be able to have access to a larger number of oocytes, but the utility of developing this technology in lower primates is much lower, and therefore this is why, after some discussion with Henry and other people, we came to the conclusion that we should rather develop this technology in the macaque, despite the inherent difficulties of the species.
I was thinking in pure biology.
Oh, in pure biology, there is no difference. I don't think there is any difference. difficulties with using pluripotent stem cells in transgenesis also hold true for rabbits, for bovine, for livestock, and therefore there's no reason to think that lower primates would be different from higher primates. Actually, the exception is mice. Mice are the exceptions. And the reason why these technologies have been developed in mice is not only that a large number of embryos are available, but it's also because mice is a very peculiar species for embryonic development.
Is that my turn? Okay. Yeah, you said that it was strictly prohibited for humans, right, to do that on humans. But eventually, isn't that going to be a problem in the development of new therapeutics and therapeutics research? That's my question. Okay. Do you understand what I'm saying? No.
Not really.
So the fact that it's forbidden to do this kind of technology in human beings, Loic is wondering if this would mean that there would be a barrier to developing therapeutics in humans which would benefit from this.
No, I don't. What is prohibited in human is the modification of the germline. Of course, you can perform gene therapy and you can introduce any kind of foreign DNA sequence into somatic cells, and this has actually been done in the context of gene therapy, although it's not a big success. But so what is prohibited is the modification of the human germline, right? And you don't need to modify the. Well, you could think of modifying the human germline in order to cure some diseases, and then the modification will be transmitted to the next generation. But this is fiction.
Okay, on a more short-term, more short-term perspective, but it's still probably pretty long-term given the difficulties in developing the methodologies. What do you think? I don't want to ask you to divulge your plans for the future, but once you've mastered the methods for producing these transgenic animals, what do you think would be the most interesting applications with this technique in monkeys? Neurodegenerative diseases are interesting, but given the life cycle of monkeys, these are very lengthy experiments. So what would be really exciting to do once we have transgenic capabilities in monkeys?
Yeah, of course, the generation time is well, it's something like 4 years. So it's a limiting factor in these experiments. But the strategies which I presented, you don't need to have the second generation to have the transgenic animal, right? So in principle, and this is actually the case in rodents, you can infect the early embryos with lentiviral vectors, or you can inject the zinc finger nuclease into the embryo, or you can inject pluripotent stem cells into the blastocyst, whatever. And then the outcome of the experiments is obtained at birth, or even before birth, because the fetuses and the newborns carry the mutations in some or all cells, depending on the technology that you are working with, right? So it means that the duration of the experiment, assuming that we are able to perform, we are able to produce bona fide pluripotent stem cells, we can colonize the embryo, and so on. The duration of the experiments is not 4 years, or it is 6 months, for instance, in the macaque monkey. You can infect embryos with a lentiviral vector, or inject pluripotent stem cells into the embryo, transfer the embryos into the surrogates, and then after 6 months you have the newborns which carry the mutations. So this is part of your question. The other part of your question is which disease we can target. And I give this presentation in the context of creating monkey models of human diseases, but these technologies could be used, and I think that they will be used once they are available. They will be used for basic research. You can knock out or interfere with the gene function, with the function of any genes involved in brain development, and then study brain development in the fetus. And these are short-term experiments, maybe not as short as there are in mice, but they are relatively short experiments, and which could help understanding how one particular gene is important for brain development in the monkey. And as far as diseases human diseases are concerned, well, I'm probably not the best person to really to answer this question because I'm not. What I'm interested in is developing the technology. Then other people will use. If we succeed, other people will use the technologies for developing the models that they are interested in. But of course, the most interesting, not the most interesting, but the easiest would be to select a disease which is induced by a single monogenic diseases. And obviously, among neurodegenerative diseases, this is a rare case. I think the Huntington disease is the only example I know. But in other diseases, such as diseases of the pancreas or the liver, there are some, or the what's called in English, the mucoviscidose. I never know in mucoviscidose. I don't know in English how.
I don't either. No.
Okay, then there is one single gene which needs to be mutated. So there are a number of human genetic diseases which can be produced, which can be created in monkeys, and for which there is a growing interest from big pharmaceutical companies, for instance.
Yeah, I mean, I've heard from several sources. I mean, in China, they treat this as a national program to produce transgenic animals. Have you heard anything about, and in Japan as well? But do you have any more recent information?
No recent information. But yeah, indeed, the China, because of the availability of monkeys in southern China, there are huge investments in these technologies. Yeah. Definitely, including investment from the NIH in China.
Right. Well, thank you very much.